Hello, you are welcome back to session 3 of Unit 5. Under this session, we will look at how nerve impulse is generated, which is one of the properties of excitable tissues. And you will also be introduced to some terms such as resting potential, action potential, depolarization, and repolarization. By the end of this session, you should be able to explain the term membrane potential and also list the stages of an action potential and describe the stages of action potential and also explain the occurrence of hyperpolarization in the generation of action potential. Some tissues are made up of cells that are able to generate or conduct action potentials. An example is the neuron and muscle cells. And the neuron cell respond to stimulation by producing action potentials as a means of communication, whereas the muscle cells either generate or respond to action potentials by contracting. The plasma membrane is known to be a bilipid layer with proteins that span the entire membrane, referred to as integral proteins. And these proteins provide small water-filled channels or pores that allow the transport of certain water-soluble substances. So transport of ions across the membrane, for instance, the sodium-potassium exchange pump, requires active transport, and this active transport also requires the use of energy in the form of ATP. So for a membrane potential to be generated, we have to understand that the extracellular fluid around the neuron has a higher concentration of sodium in it than the intracellular fluid, and is due to the sodium-potassium exchange pump, which transports 3 sodium ions out of the cell in exchange for the import of 2 potassium ions into the cell. Although the sodium-potassium pump works continuously, sodium ions are mostly returning into the cell and potassium ions are leaving it. However, the membrane is more permeable to potassium ions than to sodium ions. So the amount of potassium leaving the cell is greater than the amount of sodium entering it. Because of this, there is a net negative charge on the inside of the cell membrane and a net positive charge on the outside. In this situation, the membrane is said to be polarized. We say that the membrane is polarized because there's attraction of unlike charges, and there is the generation of electrical potential across the cell membrane. And this potential is also referred to as membrane or resting potential, and it's present in all cells. However, for excitable cells, they are able to change their potential in a predictable and controlled manner. Now, one may ask, what is an action potential? And so we will look at action potential in the next slide. An action potential indicates the sudden disturbance in the polarized state of a membrane when it responds to a stimulus. And so when a neuron, which is an excitable cell, is not being stimulated, its resting membrane potential is about minus 70 millivolts. And during an action potential, the polarity of the membrane reverses, and it occurs in such a way that the inside of the cell becomes more electropositive than the outside before falling back to its resting level. And this changes as a result of a sudden increase in the permeability of the membrane to sodium ions. Action potential occurs in 3 stages, and these are depolarization, repolarization, and returning to the resting membrane potential. Now let's have a look at depolarization as one of the Stages of action potential. When a factor causes the membrane potential to become less negative, we term it as depolarization. And some of the integral channel proteins that are embedded in the membrane of an axon are of a specific type. And some of these channel proteins include the voltage gated sodium channels. And these channels, when they open, when the membrane first begins to depolarize following a stimulus, there's movement of sodium ions into the cell down their concentration gradient. And this movement of sodium ions into the cell further depolarizes the membrane, causing more sodium ions to move into the cell. So at the height of action potential, most of the available sodium ion channels are open and the inside of the cell becomes more positive. This has been illustrated in this particular figure by plotting the change in membrane potential over time. So the influx of sodium ions takes the membrane potential from a less negative depolarizing phase to a positive one. The second stage of action potential is repolarization, and repolarization is somehow opposite that of depolarization. So for repolarization, it occurs when the sodium channels become inactive, and as a result, the voltage activated potassium channels in the plasma membrane opens. And this causes the potassium ions to leave the cell down their concentration gradient, and the membrane potential trying to near its resting stage results in the gradual obtaining of more electronegative state. The resting potential is rather resumed when the potassium channels are closed and the sodium channels move from an inactivated state to a closed position. Indicated in this diagram is the membrane potential, which becomes transiently more electronegative than it was at the start of the action potential. And this is because the relatively low closure of the potassium channels and the membrane is said to be hyperpolarized at this point. Following this, the membrane potential is therefore restored. The third stage of action potential is return to resting potential. This is where the action potential or the resting potential of the membrane is restored. And when there is a passage of an action potential along the membrane of an excitable cell, such as the nerve cell, it leaves the cells with slightly more sodium ions inside, whereas there is more potassium ions outside the cell. So the sodium potassium pump helps to return the membrane to its resting potential by pumping 3 sodium ions out of the cell and causing the influx of 2 potassium ions. The sodium gradient that is re-established indicates that the cell membrane is then prepared to generate a fresh action potential. And to generate an action potential, an excitable cell needs to be exposed to a stimulus. So in this case, a nerve cell is supposed to be exposed to a stimulus of a certain minimum strength. And this minimum strength required is referred to as the threshold. So a stimulus below this threshold will not bring about an action potential, as the axon does not depolarize sufficiently to allow the voltage-gated sodium channels to open. Now have a look at these trial questions. What is an excitable tissue? What is an action potential? What is the membrane potential? List the phases of an action potential. And then also name two ions associated with the generation of an electrical impulse. Under session 3 of Unit 5, we studied how the nerve is able to convert external stimuli into electrical signals. And these electrical signals are messages that can be transmitted from one point in the body to another point. And you got to understand that it is one of the properties of the nerve and other excitable tissues. You also learned in detail what is meant by resting potential, action potential, depolarization, and repolarization.